Dang these useless blood-sucking mosquitoes. They're not good for anything.
But wait, they may be useful.
Mosquitoes carry a lot of diseases and parasites, including malaria.
Also known as Plasmodium.
Now there have been many attempts to reduce malaria transmission through netting, pesticides, etc., but the key may instead be the mosquitoes themselves.
To reduce the spread of malaria, researchers are looking to replace natural wild-type mosquitoes with genetically modified mosquitoes. These genetically modified mosquitoes would have increased resistance to Plasmodium, making them less likely to carry and transmit malaria. Now, this sounds like a great idea, so why hasn't this been implemented yet?
Well, there are fears that the GM mosquitoes would not be able to survive and compete in the wild, which is the further release of the strategy. In this paper, the plausibility of the solution will be discussed. Okay, before we continue, let's clear up some confusion you may have. GM refers to genetically modified mosquitoes, whilst WT refers to wild-type mosquitoes.
As a whole, the research focuses on seeing whether GM mosquitoes have the fitness to survive and compete with WT mosquitoes, to see if introducing GM mosquitoes into the wild would be a viable solution to controlling malaria.
The researchers compared the lifespan of the genetically modified mosquitoes to the wild-type mosquitoes by giving the mosquitoes blood meals and compared the fecundity of the mosquitoes by observing the number of eggs laid after gonotrophic cycles. These results showed no significant differences between the lifespan of female GM and wild-type mosquitoes, and no significant difference in the number of eggs laid between them either. The researchers then took the two most resistant genetic modifications and set up an experiment where genetically modified and wild-type mosquitoes would interbreed to see if genetically modified mosquitoes were more competitive. Under Hardy-Weinberg conditions, it was calculated that genetically modified mosquitoes would represent 75% of the population. In the actual experiment, genetically modified mosquitoes actually made up 90% of the population, showing that there was some sort of advantage given to the genetically modified mosquitoes, making them competitive. However, this trend only appeared in mosquitoes that expressed the genetic modification in their midgut. This led to researchers asking why genetically modified mosquitoes were more competitive. After a few experiments, they found out that it was because of differences in midgut bacteria. The genetic modification would increase the innate immune system, causing lower levels of bacteria or microbiota in the midgut. This was found in an experiment where two groups of mixed mosquito populations were bred. One of the two groups was heavily exposed to antibiotics. It was found in the group with antibiotics, GM mosquitoes made up 75% of the population as Hardy-Weinberg would predict, but in the other group, GM males preferred to mate with wild-type females, and GM females would prefer to mate with wild-type males. This gives GM mosquitoes a sexual advantage and explains why they are more prevalent than Hardy-Weinberg would predict.
To test the stability of malaria resistance, mixed populations of genetically modified mosquitoes and wild-type mosquitoes were observed. After 10 generations, the resistance of mosquitoes was measured. Here the data is displayed. It can be seen that the genetically modified mosquitoes maintained resistance even after having mated repeatedly with the wild-type mosquitoes.
This figure here summarizes the interactions between genetic modification, the innate immune system, the microbiota, and the mate preference of the mosquitoes. The genetic modification affects the innate immune system by making the mosquitoes more resistant to Plasmodium. However, altering the innate immune system also alters the bacterial resistance of the mosquitoes. This alteration to the bacterial resistance affects the microbiota composition found in the GM mosquito's midgut. The research also found that mate preference was affected by the composition of the microbiota in the mosquito's midguts, meaning that the genetic modification altered bacterial resistance, which altered the microbiota, which alters the mating preferences found in the GM mosquito populations. This alteration to the mating preferences gave the GM mosquitoes a sexual advantage, and in turn leads them to being more prevalent in the population.
In summary, the research paper suggested that genetic modifications of the GM mosquitoes made them more competitive in the experimental population. However, the data is in a controlled setting and would be different in the wild. Studies have shown that different microbiota are in the lab versus in the wild. Therefore, we would need further testing in a field setting to verify these results.
The use of genetically modified mosquitoes to reduce the spread of malaria should be further pursued and investigated due to the stability of the genetic modification, the ease of making the resistance more complete by using multiple or stronger genes, and the likelihood of the genetic modification to spread due to modified mating preferences.